So in this video, I'm going to describe to you my recent work on microRNA biogenesis that just came out in Molecular Cell last month. I'll tell you upfront that the gist of the paper is the following. MicroRNAs are derived from hairpin-like structures, but some of these hairpin structures are not optimal for processing. We find that in mammalian cells, the processing of such suboptimal hairpins can be enhanced if it is linked to an optimal hairpin. And this phenomenon, which we call cluster assistance, requires a small protein called ERH. First of all, just a brief introduction to the microRNA pathway. So microRNAs are these short, about 22 nucleotide RNAs that together with the organelle protein can repress mRNA expression. Cells have a dedicated pathway to make microRNAs, and what I've been focusing on is the initial processing step from the primary microRNA transcript to the pre-microRNA hairpin. And this is carried out by the microprocessor complex consisting DROSHA, the endonuclease, and DGCR8, the cofactor. The initial question was, it's very easy to come up with a hairpin in the transcriptome. How come we only have slightly over 500 microRNAs? We think that the microprocessor is the gatekeeper. And so then the question becomes, what are the specific features that microprocessor recognizes on bona fide prime microRNAs? Over the years, we've learned a lot about microprocessor specificity. And so here's a summary of the features that define microRNA hairpins. In terms of secondary structure, it is a stem loop structure with an optimal stem length of 35 base pair. Microprocessor also prefers an apical loop larger than 10 nucleotide and also needs a single-stranded flanking region for processing. There are also primary sequence features that help orient and position microprocessor for cleavage at the right place. What I want to note is that you don't have to have all the features to become a microRNA. And so we call this a manual of features, meaning we can choose a subset of these features and still be able to make a. However, some microRNA hairpins seem to have really defective structures. For example, this microRNA miR51, it is a microRNA important for erythropoiesis. It has a very short stem of 31 base pair, an extremely short loop of only 4 nucleotide. And yet, this is the almost the most abundant microRNA expressed in erythrocyte. Another microRNA miR181B-1, important for immune system, also has a short stem of 31 base pair. And so how are these naturally occurring defective hairpins recognized and processed? I noticed that both of these defective microRNA hairpins reside in microRNA clusters, meaning there is another microRNA hairpin not too far away from them. And so the distance between these two hairpins are usually around 100 to 500 nucleotide. And these neighboring microRNA hairpins seem to have optimal hairpin structure. And so could it be that these neighbor hairpins can somehow help these defective microRNA hairpins for processing? So to test this possibility, I did the following experiment. I ectopically expressed the microRNA either by itself or in a cluster context. In this case, I expressed the miR144-451 cluster in HEC293 cells, where this cluster is normally not expressed. I also used a bidirectional promoter to ectopically express miR1 as a control for subsequent northern blot analysis. If I overexpress miR144 by itself, we're looking at mature microRNA accumulation on a northern blot here. You can see really robust expression of miR144. If I overexpress miR51 by itself, the accumulation is really weak. However, if I link these two microRNAs together, so co-express them as a microRNA cluster, then you can see a huge boost in terms of miR51 accumulation. So it's 40-fold higher than when it's expressed by itself, whereas the level of miR144 did not change. I want to note that you see multiple bands for miR51, and that's because of its non-canonical biogenesis downstream of Drosha, namely ago slicing followed by PARN trimming. Now, if I supply miR144 on a separate strand of RNA, expressing it downstream of miR1, then we lose the enhancing effect. This suggests that the increase in miR51 level is not a regulatory effect of the mature miR144. Rather, what we call the cluster resistance is a cis effect. Now, if I switch the order of the two hairpins, I can still see the cluster resistant effect. However, if I now link two miR51 together, we don't see the enhancing effect, suggesting that we really need an optimal MicroRNA hairpin to be the helper hairpin. So to test whether the cluster resistance depends on the recognition of the helper hairpin by the microprocessor complex, I then mutated miR144 to change its optimality. And so the stem length of 144 in the wild type is 34 base pair. And if I shorten it by 5 base pair, now miR144 is suboptimal, its accumulation decreased, and that also decreased miR51 accumulation. I can then introduce a compensatory mutation to restore the stem length of 144. Now you can see the expression of both microRNA are restored. I can also make miR144 suboptimal by lengthening the stem. So now making the 34 base pair 39 base pair. You can see the level of miR144 went down, and so did that of the miR51. I can also rescue the effect by making the 144 stem optimal again. And so you can see the level of 51 really tracks with that of 144, which is in turn a function of its optimality in terms of microprocessor recognition. And so I conclude that cluster resistance must somehow involve the recognition of the helper hairpin by the microprocessor. I then explored the distance between the two microRNA hairpins. So in the wild type, the distance between the miR144 and 51 hairpins is 95 nucleotide. Now if I increase it to even up to 900 nucleotide, I can still see robust cluster resistance effect. Even at 3000 nucleotide, the expression of miR51 is still higher than when it's expressed on its own. And so we conclude that cluster resistance can operate over widely spaced hairpins. So far, I've only showed you plasmid-based transfection experiment. Now I'm going to see if this effect holds in endogenous microRNAs. And so for this, I turn to K562 cells where the miR144-51 cluster is normally expressed. I then used CRISPR-Cas9 to introduce small deletions and mutations in either the 144 hairpin, the linker region, or the 51 hairpin. I then derived clonal cell lines of these mutants. You can see compared to parental cells, if I introduce mutations in the miR144 hairpin, I can see a loss of 144, but the level of 51 also decreased significantly. If I introduce mutations in the linker region, neither microRNA are affected. And if I introduce mutations in miR51 hairpin, you can see miR51 is gone, but the level of 144 did not change. And so this is consistent with what we found using the plasmid-based assays. And so cluster resistance can act on microRNAs expressed from their endogenous loci. So far, I've only showed you Mature microRNA is an assay, which is a function of both microRNA biogenesis and microRNA decay. To more directly test prime microRNA processing, I then use this in vitro cleavage assay where I in vitro transcribe the RNA substrate, label it at 3' end, and then monitor the cleavage product over time. And so this is cleavage of prime miR51 on its own. I can also assay for miR51 cleavage when it's linked to 144. And so here we can actually see a cleavage intermediate where miR144 is cleaved first. So using this assay, I can then derive cleavage rate constant for miR51. And so you can see here when miR51 is linked to 144, the rate constant is more than tenfold higher than when it's on its own. And so this really suggests that cluster resistance acts at the level of prime microRNA processing. And finally, I want to test if this phenomenon is more general. So in this case, I expressed miR181B-1. This is also a defective hairpin with short stem. I express it on its own or link it to another optimal hairpin, miR125A. And so again, you can see a huge boost when 181B-1 is linked to 125A. And so this suggests that cluster resistance can enhance the processing of other microRNA hairpins, and it's not restricted to miR144 51. So then the big question is, how does it work? So in the in vitro cleavage assay, I initially used a lysate that overexpressed microprocessor complex. So that left open the possibility that some additional lysate factor can contribute to this effect. And so what I then did was to use purified microprocessor. And in this setup, I can see that miR51 cleavage was not better when it's in a cluster context. If anything, it's worse. However, if I then supply the purified microprocessor with a lysate that comes from a cell line that has microprocessor deletion, then I can restore or recapitulate the cluster resistance effect. And so this really suggests that there is additional factors that are involved in this cluster resistance effect. Now, in fact, a few years ago, I actually co-purified with microprocessor a small protein called enhancer of rudimentary homolog, or ERH. And so in this purification, I first pull on DROSHA with a flag tag, and then load this microprocessor with a prime microRNA substrate that's labeled by distal biotin. And so using this strategy, I can have a really clean prep of the microprocessor complex. And ERH is the only protein that comes down in this prep. And so this actually suggests that ERH stably binds to the microprocessor, and maybe it's actually a bona fide component of the microprocessor complex. Now back then, we didn't know about cluster resistance, and I had a really difficult time assigning a function of this small protein. Now that we know the cluster resistance, could it be that this small protein is actually more specific for cluster resistance? And so this is what I'm testing here. So I knocked down ERH using CRISPR interference. There are two independent guide RNA knockdown in K562 cells. I then use qPCR to amplify across the hairpin, so assay for the precursor hairpin. If a microRNA processing is inhibited, then you would expect accumulation of the precursor. And that's exactly what we see for miR51. And so when in these two knockdowns, the level of the precursor miR51 hairpin increased about 15-fold, whereas the 144 hairpin cleavage is not affected. And so ERH knockdown is more specific to miR51, consistent with our hypothesis that it's functioning in the cluster resistance. I could also look at mature microRNA accumulation by small RNA-seq. And in this sequencing experiment, miR51 is also the most significantly affected microRNA when I knock down ERH. And so we conclude by this experiment that ERH enables cluster assistance. So this is basically where we are at right now. We don't know how exactly ERH works to facilitate cluster assistance, but I'd like to put forward some working models. So first of all, I want to note that Sebastian Herzog's lab in Austria also reported the same phenomenon. And in their paper, they identified through CRISPR screen another protein called SAFB. It's a nuclear matrix protein. Seems to be also important for cluster assistance. And so because ERH and SAFB can dimerize and these two proteins can interact with each other, it's possible that these interactions would actually mediate dimerization or even multimerization of the microprocessor complex, such that binding of the helper hairpin by one microprocessor can then promote binding of another microprocessor to the recipient hairpin. Another alternative model is what we call a transfer model, where the microprocessor monomer, after cleaving the helper hairpin, is then transferred through some sort of partial bound intermediate state to the recipient hairpin. And we're not exactly sure how ERH might be involved in this model. I also want to note two relevant papers that report on similar findings, one by Eric Lai's group and one by Nara Kim's group. To summarize, we find that some natural microRNAs are embedded in these defective hairpins, which won't be efficiently processed by the microprocessor complex on its own. However, if we link these defective hairpins with a helper optimal microRNA hairpin, then they can be processed efficiently. And this seems to depend on the small protein that co-purifies with microprocessor, and that protein is ERH. And with that, I would like to thank the Bartel lab, especially Dave, for his support and guidance, everyone for helpful discussion, the White Hat core facilities, and also the funding source. Thank you for listening.